It's my pleasure to introduce our next speaker, Ian Charest. Ian Charest is an associate professor in the Department of Psychology at Université de Montréal. His research focuses on how the human brain processes visual information using neuroimaging techniques like fMRI and imaging. He also investigates cognitive processes such as perception, memory, and decision-making through advanced computational modeling methods, including machine learning and representational similarity analysis. Ian Charest holds a Courtois Chair in Cognitive and Computational Neuroscience, which is supporting the development of an online platform for the cross-disciplinary investigation of behavioral, computational, and neuroimaging datasets. Join me in welcoming Ian Charest.
All right, well, thank you for the kind introduction, and thanks to the organizing committee for inviting me at this edition of Maine. So my talk is about visual representations and how they are predicted by large language models. So as was said a minute ago, in my lab we study vision in the human brain using a collection of tools and techniques, including neuroimaging, psychophysics, and computational modeling or artificial intelligence. And today I will present some of this work that we've been conducting recently, starting with the natural scenes dataset, some work using large language models to predict visual responses in the natural scenes dataset. And finally, I'll show a real tour de force led by Peter Brotherwood, who's here today, where he's collecting massive electroencephalography data that is a replica of the natural scenes dataset, but with time. So what is the natural scenes dataset? This project was led by Thomas Naselaris and Kendrick Kay. The data was collected at the University of Minnesota. And it's a massive data set consisting of high-resolution brain responses to over 70,000 natural scenes. And these scenes are all paired with human-generated captions and annotations. And so this data set really has all the correct ingredients to test novel computational models and hypothesis spaces that we couldn't really test before because we didn't have the data to do it. So when we see this visual scene, we know that the human brain extracts a lot of complex information from this visual input. It could include finding the objects like the kayaks. It could find a sailboard, their spatial and semantic interrelations, and their interactions with the environment, like the windsurfer navigating the lake. However, coming up with a quantitative approach for studying this information can be challenging. In the next few slides, I'll show you how we tested whether the contextual information encoded in large language models could help us better capture the richness of this natural scene. Large language models are trained on massive data sets of billions of tokens, pretty much everything we've ever written and digitalized. And the scale of the textual information, we think, leads these models to converge on a decent characterization of the structure of the world. And today, I want to make the bold prediction that this is exactly what sensory systems also do. So to test whether the representational format on which these LLMs converge is useful to make predictions about brain responses to natural scenes, we're going to use two complementary approaches. So each stimulus is paired with high-resolution 7 Tesla fMRI activity, and with human-generated scene captions, like these 3 women playing frisbee in the field. And these scene captions can be passed through a large language model. We can also look at the brain activity and looking at activation patterns or voxel responses. And we're going to model the activity patterns or the voxel responses using representational similarity analysis. We can apply representational similarity analysis to the latent spaces where the scene captions land into the large language models. And we're also going to be using voxel-wise encoding. I'll show you examples of both these techniques. To capture the representations from the scene captions, we will use MPNet, which is a sentence encoder. And at the time where we started this project, it was the leading sentence level encoder on Hugging Face. So I'll show you first the results using RSA. So how does RSA work? To devise a model based on the scene captions, we take these scene captions and pass them through MPNet. We compute the representational distance between their respective embeddings. So the embeddings is a point in this multidimensional space, in the latent space of the LLM, and we can compute the distance between these two embeddings. And we can store this distance in what we call a representational dissimilarity matrix, or RDM, and we repeat this process for all pairs of scene caption embeddings, and that gives us a characterization of the representational geometry from the LLM embeddings of scene captions. To see whether the MPNet embeddings can predict brain responses related to the visual scenes, we will use RSA, representational similarity analysis, with a searchlight mapping. So from a spherical searchlight that extracts brain activity patterns from a local brain region, we can do the same thing. We compute the pairwise distances between the activity profiles and put that distance into an RDM. And now we can move the searchlight around the whole brain until we have RDMs for every little local bit of the brain. And then we can compare this searchlight RDM with our MPNet or LLM embedding of scene captions based RDM. So we can use any metric to do this, and for simplicity we use the Pearson correlation. So here's the first result. This is a cortical flat map which is obtained by cutting a cortical surface and projecting it down onto a plane. And you see the back of the brain is the calcarine sulci is what we used for cutting it, and the back of the brain would be here, so this would be early visual cortex, and you move along the ventral stream in this direction. This would be the dorsal part. Anyways, so this shows us where in the brain the representational alignment between brain responses and the LLM embeddings is mostly significant. And this searchlight result here is shown at the group level, so we averaged all the searchlight results across our 8 participants. And it's remarkably robust, and it's observed also in every single NSD participant. Okay, so I told you earlier that we think, just like LLMs kind of capture the statistical regularities of the world through the language input it receives and is trained on, we think that the sensory systems also project their inputs into a rich representational format that characterizes the structure of the world. And so we're making the hypothesis that the visual system transforms visual inputs into this high-level representation that captures the structure of the world. And we're going to formally test this hypothesis by training visual models to predict LLM embeddings of scene captions. So we trained, and this work was done in collaboration with Tim Kietzmann, who's at the University of Osnabrück, and Adrien Dorig, who's now PI at Freie Universität in Berlin. So we trained recurrent convolutional neural networks that take images as inputs and predict the LLM embeddings of the scene captions describing those input images. And they do this via nonlinear transformations and propagation to approximate the LLM embedding. We then use Searchlight RSA, just like I've showed you before, to quantify the representational alignment with our brain data. And we see, again, a very similar map of brain areas that is very similar to what we observed when we did the Searchlight, just using the LLM embeddings themselves. But the correlation is significantly larger compared to the model where it was just based on LLM embeddings of the scene captions. This, again, is a robust result that is observed in every single NSD participant. Okay, but we also ran a control analysis, highly controlled model comparison that contrasts our LLM-trained RCNN with a RCNN that has exactly the same architecture and the same parameters, but is trained to predict the category labels that are contained in the natural scenes. And to adjudicate between the two models, we will contrast the resulting Searchlight maps. Here, I plot the Pearson correlation difference in model to brain representational alignment. And overall, we find that our LLM-trained RCNN outperforms the categorical model in most visual areas, perhaps with the exception of early visual areas where both models performed equally well. Okay, so next we performed voxel-wise linear regressions to predict voxel activities from our LLM embeddings of the scene captions. And so how does this encoding modeling business work? So we have fMRI activity in responses to these natural scenes, and in a nutshell, we try and find a set of linear weights that applied onto the MPNet embeddings of the scene captions plus some error predicts the fMRI responses. And we do this in a cross-validated fashion, which is we train on a subset of the natural scenes and we test on some held out natural scenes. And once we've found those linear weights, we can simply project them on the test set embeddings and get predicted fMRI responses. To evaluate the model performance, we simply look at the correlation between the predicted fMRI activity and the measured fMRI activity on the test set. So, yeah, just to give you flavor of the sort of representational coverage between the training set and the test set, here I'm showing a TSNE map projected on the 2D plane that shows in gray dots the location in a multidimensional semantic space of the training images, and most importantly in yellow dots the locations in the space of the test set. So the test set has a pretty good coverage of the sort of semantic information that is covered by the training set. And I'm not going to talk about this because of lack of time, but we also do decoding on this data. And so the blue dots here show how well we decode the scene captions that the participants were, well, of the scenes that the participants were currently seeing. All of this is in the preprint, so you can have a look at that. Okay, so the linear encoding revealed a similar network of brain regions compared to what we saw with Searchlight RSA. So the responses in these highlighted voxels can be predicted very well by a simple linear mapping between the LLM embeddings of the scene captions and the brain responses to the natural scenes. And again, this showed to be a strong result, and we observe it in every single NSD participant. And some accuracies reach correlations as high as 0.86, so our model is able to explain a significant, pretty large amount of variance in the data. Now the cool thing is, once we've learned the linear mapping, and this is similar to what Ale showed us this morning, once we've learned the linear mapping between a voxel's response and the LLM embeddings, we can start playing cool games. We can test whether the contextual information that is captured by the LLM model can help recover known organizational principles or classic neuroscientific findings from the vast visual neuroscience literature. For example, I can take these two example sentences, so a man with a beard smiling at the camera, or a view of a beautiful landscape, and pass them through MPNet to get their embeddings. And now I can project for each voxel the learned linear weights onto these embeddings to get predicted brain responses. Then I can contrast these two predicted brain responses. So in warm colors would be regions that respond more strongly to the sentence of a person, and in cool colors, regions that respond more strongly to the view of a beautiful landscape. And what we find when we do this contrast is that we're able to recover the known organization that has been observed for decades in the neuroscientific literature. Namely, in warm colors, the fusiform face area network, so regions that respond more strongly to faces, and in blue colors, the place selective network, so regions that respond more to places. The white contours, I don't know if you can see them, show the regions of interest that are obtained from the independent functional localizer that is also available with the natural scenes dataset. So our regions that are obtained from simply understanding the linear mapping between LLM embeddings and brain responses match pretty well with the regions of interest that are defined functionally with the functional localizer. So what this tells us is that the LLMs can encode important dimensions that are used by the brain in vision. Okay, so here's another example where we recover the food network that was recently observed and discovered by many groups, including Nancy Kanusher's group at MIT. So using sentences containing foods and contrasting that to sentences not containing foods, we again reveal known functional organization in the neuroscientific literature. So if we wanted to make a point about a general representational format, the learned mapping between brain responses and our model should generalize to brain responses in the brains of different individuals. So to test this, we use exactly the same encoding strategy, except that we trained on participants' brain responses and tested on an independent participant. And this time, the predicted brain activity comes from one person's brain, and the measured activity comes from another person's brain. And so if this works, this would reveal regions at least where the format or the representational format is general enough that it can enable us to make predictions from one's brain to someone else's brain. And there's a lot of data in this slide, but this slide is showing training on the rows on the 8 NSD participants and testing on the columns on the different, the other participants. So on the diagonal is a result that I've showed you already, training and testing on the same participant. And maybe the first thing you notice is how this diagonal pops up. And this is indicative of some kind of idiosyncratic component in the data where you can predict someone's brain responses better from their own brain than from someone else's brain. But we also find some pretty decent generalization. So if we train, for example, on participant 3 and test on participant 2, we still see this network of brain regions that we saw since the beginning of my talk. And so this is indicative that there is some generalizability, and maybe that representational format is indeed something that captures the structure of the world. So maybe one note here is that we did not use any functional alignment methods or anything. We simply projected all of the data on Free Surfer's FS average, which is this surface representation template space that comes with Free Surfer. Okay, so how am I doing in time? Okay. So the intern summary is that by training on large-scale linguistic corpora, which is thought to be crucial for learning good embeddings, the LLMs achieve something quite remarkable, characterizing the structure of the world through a wealth of contextualized and behaviorally relevant information. And this rich learning signal may provide some benefits in training our models. fMRI and more recently the natural scenes dataset have given us amazing opportunities to learn stuff about the spatial organization of scene processing, but fMRI doesn't really tell you a lot about how the neural dynamics, how is this implemented dynamically in the brain, and this is an important component even for static images. And so we went ahead and collected, we're in the process of collecting NSD-EEG, which is an electroencephalography data set with the natural scenes data set, with the images from the natural scenes data set that Peter has been working on. So you've seen this figure already. This is just showing the sort of recent large-scale neuroimaging data sets that have been produced. You have the NSD on there. I don't know if you see my mouse. Yeah, the NSD would be here. CNeuromod is outside of this room. We would be somewhere around here with over 90 hours of testing in each participant with NSD-EEG. So it uses the same 73,000 stimuli. Of course, we couldn't bring the participants from Minnesota to Montreal to do this, so it's 8 different participants. To try and compensate from more noisy EEG signals, we upped the number of repetitions to 10 trials per stimulus. And this leads us to over 100,000 trials per participant. We're using a high-density 160 electrode EEG system, and we're also collecting 3 Tesla fMRI data on a subset of the images in the same participants. We're measuring everything in a semi-anechoic Faraday cage, which helps mitigating some noise that might be in the environment. And here's an example of a semi-happy participant. So we selected these 8 semi-happy participants by carefully looking at the brain activity of a set of initial 25 candidates. We got these 25 people to come to the lab and scan them using 300 independent stimuli sampled from the Things dataset. And we selected them on the basis of a combination of global SNR, ocular artifacts, how well they were performing the task to try and maximize the value of this data set by selecting the best 8 participants. To give you a flavor of the data quality, we looked at test-retest reliability, and I'll skip the details in the matter of time, but we looked at how well random split half analysis on the image level would do. And so we simply look at 2 different averages of independent subsets of trials for an image and correlate the topography at each millisecond. And then we average across all of the stimuli. And here is the result. And we see strong correlations. And we see that before the stimulus onset, which would be at 0 millisecond here on this plot, the correlations are flat and non-existent. And then we have this sharp rise in correlations at around 70 milliseconds that persist until the end of the trial, pretty much. We also tried the same approach of encoding, but this time using the EEG data. So it's the same idea. We try and find a set of linear weights that, applied to the LLM embeddings of the scene captions, predict the electrode amplitude, the channel amplitudes, for a given time point. And then we can look at the similarity between the predicted channel amplitudes and the measured ones. And again, we see very decent similarity, so very decent performance of our model with temporal dynamics that makes sense given the nature of our trials. And this is remarkable given that at the moment when Peter generated this figure, we only had 30% of the whole data collected. So in some cases, some participants now have completed at least the EEG part of NSD-EEG, and we expect these numbers to go up when we have 100% of the data. Okay, now that we have these extensive, massive data sets with the same images in fMRI and EEG, the next challenge is to see if we can combine the 2 data sets in a way. And we're working with Ale and Radek Chihe to try and develop new methods to fuse fMRI and EEG data. And so to do so, we're going to try and predict voxel responses using a linear combination of the EEG channels at every time point. So again, we try and find a set of weights that, applied to the EEG channel amplitudes, predict the voxel's responses. And then we try and get a measure of accuracy by comparing the predicted fMRI activity with the measured fMRI activity. And if we repeat this for every EEG time point, we think this produces an empirical spatiotemporal source reconstruction technique. And so we can look at this result in a movie where you're going to see the predicted brain activity in time from minus 50 milliseconds, so 50 milliseconds before the stimulus actually came on, all the way until the end of the trial. And if I play this movie, initially we don't see anything, and then around 70 milliseconds, the back of the brain lights up, so the early visual cortex lights up. Let me play it again. Let me pause it. Right, so we capture early visual processes at the beginning, which is a good sign. And then if I leave the movie playing, we start capturing later processes. We see this traveling of the information along the ventral stream and dorsal streams. And maybe the most important part, at least for my talk, is that this spatiotemporal information processing mapping that we observe lands on this set of brain regions that we identified with our LLM embeddings of scene captions since the beginning of my talk. Okay, so I'll conclude. I hope to have convinced you that vision representations in the brain can be predicted by LLM embeddings of scene captions. We think that this is because LLMs, having been trained on that amount of billions of text tokens, can capture the structure of the world, and that this is also what sensory systems do. We think that combining RSA analysis and voxel-wise encoding modeling provides complementary evidence. RSA is parameter-free and perhaps good for adjudicating between models, whereas linear encoding approaches allow further investigating the model dimensions via the learned linear weights at the cost of poor model adjudication because there's so many trainable parameters. And we think that NSD-EEG will allow further investigating of how these representations are established with unprecedented spatio-temporal precision, especially when combined with NSD-fMRI. And special thanks to everyone on this slide and my lab, the funding bodies. And if you're interested in NSD-EEG, you can register your interest by scanning this QR code with your phone, and you can tell us why you're interested and so on and so forth, and we can talk about maybe early access, or at least we can tell you more about the data set. And thank you for your attention.
Thank you so much. I think we can all agree that it was impressive, especially the last part. Question time? Yes.
Hi, that was amazing. Great talk. I'm curious, I guess one of the strengths of what you show is the fact that you're using as target embeddings from large language models that are strictly trained on text. So they're basically blind. They don't have any sort of visual spatial awareness. But I guess going back to the previous talk, which was also fantastic, I was wondering what you think could be gained, perhaps, given the importance of head motion and head movement, to perhaps train on a large ANN that's multimodal, that includes vision and language, so from the target point of view, but also from the input point of view, in a perfect world where we don't have to worry about head movement restrictions and signal noise, what you think might be gained in terms of representations in the brain?
I think a lot would be gained. So there isn't any data set available right now that has the same scale at what LLMs can be trained with. For instance, if we had, I don't know, video data of everything that was ever, done on this planet, and we could train models to predict the next frame or something, this might bring us to this kind of structure of the world characterization that we get from LLMs. And similarly, if we had embodied agents that moved around and that could capture multi-sensory information about the world, this would give us training data that would be even richer than what we have now. The short answer is it would help a lot. The other part of the answer is we don't have this data yet, and until we do, LLMs bring us a long way to try and predict brain responses.
Thank you.
Okay, we have time for another question, maybe a third one if it's very, very quick.
I'm curious to hear you say more about why the LLM embeddings were similar to the pattern of the representations of the brain. Like you said, it was because they capture the structure of the world, but I wonder if what would it mean if it wasn't aligned? There's probably a lot of constraints in the biological system, and also the LLM is trained on the way we talk about the external world as well, and how we process objects in the visual scene, in the ways that we would caption them. So I don't know. I guess partially I'm saying I don't really buy that it's just because because both of them optimally capture the structure of the world. And I'm just curious to hear you refute that or say more about what you think.
So I think part of what you said in your question is actually true. We describe the world that we see in the text that we write, right? So this information is available at training for these large language models. And so they're agnostic to visual inputs, but they're not, given that many books have been written with 4-page descriptions on the frame of a door, especially in the French literature, when sometimes it gets lengthy. So I think there is a lot of information that these models learn in training that can help generalize and predict these visual responses. And I do think that part of how a system transforms the information into a representational format that can be readily accessed. Again, like maybe it's an answer of energy, and it's only the beginning of an answer, but we think that if sensory systems encode information from their inputs into a shared representational format, that would also allow for easier integration of the information between the senses. So if you have a downstream neuron that is a multisensory neuron or something, it wouldn't really make sense for that neuron to have to change posture to read the auditory cortex and then change posture again dynamically to read the visual cortex, because these two cortices are speaking different languages. If both these sensory systems project their representations onto this sort of representational format that captures the structure of the world, then you can combine the information with simple, energy efficient ways, like adding them or averaging them or something. So we haven't done this research yet. There's a preprint that came from the lab of Rufen van Rulen in France, that shows that when people are reading sentences and when they're seeing, you can actually decode everything about them. So, it's a start, like we're kind of at a starting point of what I think is a paradigm shift, but there's a lot more work to be done before we can, close the coffin. Yeah.
Okay, I'm afraid we are fashionably late, so I'm sure you can catch up later. So we can give space to the other speakers. Thank you so much.